First, we're going to do a quick overview of their generalities, their basic structure and cells type, their ecology and feeding, reproduction, and defense mechanisms. And later, we're going to be speaking about the bioinspiration that they bring to us. So the sponges, they're aquatic organisms, and even though many people confuse them with plants, they are, in fact, one of the eldest animals of the animal kingdom. And there are some fossils dating back to the Precambrian times. So this is around 650 million years ago. And they are quite intriguing creatures because even though they don't have a circulatory system, a nervous system, or a digestive system, it makes us consider why we continue considering animals. Well, first of all, because they lack cell walls. That's the typical feature of plants. They are not able to make their own food, so they are heterotrophic. They're multicellular, and they produce sperms. And as we spoke our last presentations, there are 3 classes of sponges. And they are characterized mainly by the composition of their spicles. The spicles are the structural elements that make the structure of the sponge. And well, we have the class Calcarea, that they have spicles composed of calcium carbonate, and they don't have spongin. Spongin is a protein very similar to the collagen. So when you touch it, it's fluffy. But well, this one they don't have, so they are hard as rocks. Then we have Demospongiae, that they have silicon dioxide spicles. They can have spongin or a combination of both. And then we have the Hexactinellida. These are the glass sponges, and they are composed by silicon dioxide. Afterwards, Leonel is going to speak about very interesting bioinspiration of this class. So, ecology, well, they are aquatic organisms, and they are freshwater species. There are around 150 species that lives in freshwater, but they are mainly marine species. So they can be found in tidal zones up to 8,000 meters deep. They are in tropical rainforest, they are in tropical waters, in temperate waters, and in polar waters. So the Demospongiae and the Calcareous species, they prefer to be in shallow waters that are non-polar. And then the glass sponges, they're very common in polar waters and in very, very deep waters. So the sponges that are in temperate waters, they are more abundant, but they are less diverse. Then regarding to their feeding, well, sponges are filter feeders. So here we can see in the diagram, we have the pores that are called ostium or ostia, where the water enters. And here we can see some cells. We have these ones that are called choanocytes, that they have this flagellum, so they're going to be generating the water current. And they're going to be filtrating all the food particles that are in the water. And then the water will be going out through the osculum. So they're mainly filter feeders, but there are some sponges that they host microorganisms that they are photosynthetic. So they're going to be generating a kind of symbiosis. And then in very poor environments, there are some sponges that have evolved to be carnivore, and they can prey small crustaceans. So here we can see the structure of the sponges and the cells type. So very quickly, this is the most basic structure, but not the most common. But for pedagogic reasons, I choose this one. So the water is going to be entering the pores that are called ostia, as I told them, and then they're going to be, there is these cells that are called choanocytes that are going to be filtrating the food particles, while the water is going to be leaving by the osculum. Then we have the sclerocyte, that are the cells that are going to be generating the spicles. The amoebocyte, that is going to be delivering nutrients, and then they can differentiate to different types of cells. Then we have the porocyte, that are the one who control the osculum. The oocyte, that is the egg cells. The pinacocyte, that is the cells that are in all the external surface of the sponges, and they can phagocyte larger pieces of food. And then we have the lophocyte or cholenocyte, that are the ones who are secreting collagen or the spongin. And well, so that is all the cells type.
So now the reproduction system of sponges. So there is 3 types of reproduction. Most of sponges are second shell hermaphrodite. So they can emit, the male can emit in the seawater sperm cell that are transmitted by the current through another sponge. And the sperm cell enter in the sponge. It produces a larvae and it develops, and it at that time emit in the seawater. And so the larvae is in the current, and when it found appropriate substrate, it can develop. Another aspect is the regenerating from fragment. So small part with the right type of cells can regenerate in other parts of the seed. And the last one is by budding. So budding in French is bourgeonnement. So some of them can do that with unspecialized cells, if they find the good condition. So why sponges are so interesting? Because they have a lot of defense mechanism. We will see two types of them. One is more mechanical, because they are able to produce spicle, as Margareta said, and that spicle are not really digest, so first. And they are able to emit, to shade that spicle, to form a dense carpet to avoid the predation of echinoderm. So we can see on the picture, we can't really see the carpet, but we can see that echinoderm can't enter in the closed area of the sponge, and that's a good illustration. Only nudie branch can co-opt with the toxin that the sponge is able to produce, because I forgot to say, they produce secondary metabolite, which protects them from all the microbial and virus and other predators that are in the water. And they are able also to produce toxin that prevent them to be overgrown by other sessile organisms. So we will present you why marine sponges as a drug tracer. And first, it's a small introduction to metabolite, and then we will see three different applications of its pharmaceutical application. So first, sponges can live up to 5,000 years. So it's an animal, a filter feeder. They are living in an environment with a lot of microbes, of predator, and they are able to live until 5,000 years old. So why? So it's mainly the aspect of the secondary metabolite. They are mainly, some of them are produced by the sponge, some others are produced by symbiose with fungi and different organisms. And we can see that so this is the total number of new compounds from sponges in function of their bioactivity. The new compound found in sponges, and many of them have cytotoxicity activities. A lot of antibacterial, others are antifungal, and a lot of miscellaneous. That's why marine sponges are the main provider of new compound for marine natural products. So you have even before cnidarians. They are the first. So we have different applications, and Margarita will present you.
Well, the first compound I will present you is vidarabine. It is also known as ARA-A. And well, first I will start with a little bit of history. In 1950, there were found these two compounds that came from this sponge that is called Tetia crypta, that is a Caribbean sponge. And well, these two compounds, that is spongotimidine and spongoridine, there are two nucleosides. And well, they gave a new generation of modified nucleosides that were synthetic, made inspired of these two. That one is ARA-A and the other is ARA-C that we will see later. And why this is important? Because until 2004, Lionel told you all the amount of compounds that were discovered, but until 2004, ARA A and ARA C were the uniques that were in clinical use. So this for relativize the to have the relationships, no? So vidarabine, it is used as an antiviral agent against herpes and varicella. And here I have a representation of the mechanism of action against the herpes simplex virus. So here we can see the virus. Well, you're far away, so it's not easy to see, but we have here the capsule, and there have some structures that are like hooks. So this makes the virus very site-specific, so they can recognize the cell host, and they will attach first, and they will, by different mechanisms, they will penetrate the cell host. And once they're inside, they're going to make a process that is called uncovering or uncoating, where the capsule is going to dissipate, and they're going to release all the genomic. Well, there are two types of virus, DNA or RNA virus. This is any DNA. So the viral DNA is going to enter the nucleus, and basically what they are going to be doing is to use all the DNA replication machinery that has the cell host to replicate their own genome. So vidarabine, what it's going to do is going to inhibit the DNA polymerase. So in this way, he's going to avoid that he replicates. So well, this is the first example. Well, the second example is citarabine, that is the brother of ARA. So it comes from well, these compounds as well. And ARA is treated against leukemia and lymphoma. These are two blood cancers. So how they work? Well, here we have the representation as well. And I told you, well, the citarabine is a nucleoside. So what it's going to be doing, the citarabine is going to penetrate the cell host, and he's going to be quickly phosphorylated. And so with this new structure, his shape, his form is very, very similar to the cytosine. Cytosine is our nucleoside. So when he's going to be entering to the cell, to the nucleus of the cell, the DNA polymerase is going to think it's a cytosine, so he's going to cut it and put it in the replication in the chain of DNA that he's duplicating. And even though it's very similar, it is not the same, and this is going to block all the chain. So this is going to inhibit all the DNA duplication and transcription, and eventually it's going to induce an apoptosis that is a programmed cell death. Well, and this is going to help to stop the tumor. Then we have eribulin. This is the last compound I will show you. In the report we have more, but well, we choose some. And well, eribulin is a structural simplification of Helicondrin B. Helicondrin B comes from this sponge that is called Helichondria ocadii. And as you can see, it's enormous. This has a molecular weight around 1,000 grams per mole, so it's quite huge. So what they do here in the synthesis is synthetic. They cut this part to make it smaller, but it has the same extremities. It has the same structural shape. So eribulin was in 2010 approved to be used against breast cancer. And well, his ways of action is represented here. So here we have the microtubules. This is a network of proteins that are part of the cytoskeleton. So they're involved in many things. They have different roles. For instance, the transportation of different materials or the movement of the cell, but they are involved as well in the cell division. So here is where it works, eribulin, so that is going to be aggregating with the tubulin, that are the proteins that make this microtubule, making aggregates that are useless, and they're going to inhibit the growth of the microtubule. So when we are in the cell division, already in the G2M phase, already in the second gap and starting the mitosis, the chromosomes are going to be already condensed and central, ready in the center, and they're going to be ready to divide in the two chromatids, but this will never happen because the microtubules are totally blocked. So this is called persistent mitotic arrest, and it will induce an apoptosis as well, that is the programmed cell death. And this will help with the tumor regression. It's going to avoid that the tumor continues expanding.
So now we will present something really different from human health application. It's marine sponges as a structural engineering design. So we will present two applications, which is mechanical assembly and optical performance. So for both of them, it's the same species, Euplectella aspergillium. What is interesting? So in natural environment, resources are often limited. So the sponge has to develop in an environment without too much resources. She needs to feed, so she needs to be in a current, where there is more food for it. So this species had developed a special structure, a special skeleton made of a glass. It's silicium. It's silice, sorry. So why is it better? Because from different scale, from the nanometer scales to the macroscopic scales, they have different shape that can inspire engineers to develop a new kind of structure, because it reproduces different circle shape and assembly. And you can observe at the macro scale different circle, different square with diagonal line, and it gives to the sponge something really light, something really strong, and at the same time really flexible. So some researchers are working on it, and it can we can improve our technology with that, mainly with a 3D printer. And another aspect of this structure, it's the optical performances of this skeleton, because so it's glass. This species is found between 30 meters and 5,000 meters, so there is no symbiosis with an algae, for example. So we don't know if the optical performance of the sponge is just a coincidence, or if it's useful for the sponge. But what we know about that is that first the shape of the structure is really close to commercial optical fiber. But with some improvement, as the shape at the end, we can capture way more of light with a large angle, so it's an improvement. And as actually, when we produce optical fiber, we need a lot of energy. We need to heat it, we need pressure, so it consumes a lot. And the advantage of the biosynthesis of glass is that you can incorporate, for example, sodium in the structure. That's what we cannot do actually with our process. So that's something. Yeah, that's an innovation we can include in the manufacturing of this type of stuff. And of course, we have also the strong structure, which is also a benefit, because we can really commercial fiber are fragile most of the time, so it's an improvement also for this aspect. So we can produce low-cost solar cell by this process. So as a conclusion, what is interesting is that marine sponges have evolved through the geological time. So it's a really old organism, 650 million years. And they have to evolve in a hostile environment, and they can produce, they can be a drug tracer. They can give us a lot of innovation for manufacturing product. And also, what we think is that it's important to give them a bigger importance, because they are not well recognized. We have probably, it's important to protect them, to include them in different programs, because at this time they are not recognized for that, I think. That's it. Thank you.